So we have now 9 participants and session is already delayed, and this session is a total of 90 minutes. So I request to all participants to limit their talk within 10 minutes of time. Otherwise session will be so late. So first of all, I am inviting to Dr. Iqtidar Hussain from Pakistan. He is from Department of Agronomy, Faculty of Agriculture, Gumar University. There are Ismail Khan from Pakistan. So, sir, now you can start, and please be limit
Thank you,
10
sir.
minutes or so. Yeah. Welcome, sir. Welcome. Now, stage is yours. You start, please.
No, I am sharing my screen.
Yes, you are.
All things are visible.
It's visible. Just go to slide show mode. Go to the slide mode.
Slide mode.
So it is on the top, slideshow mode, on the top.
Enter. Share. Yes. My topic of research is.
Sir, it is not visible. Your slide is not yet visible. Yes, now it's coming. It's coming. But go to slideshow.
I am sharing. This is no clear my slide.
No, not visible yet. Not visible yet. Yeah, just coming. Now you go to the slideshow mode on the top.
देखो slide show mode.
Yeah.
No, it is clear.
It's clear, but the slideshow mode would be a rather good thing if you have. You go on the menu bar on the top, where you can see the insert, transitions, animation, slideshow. Yeah, just. Yeah, it's here. Yeah, it's here. So now from beginning. Yeah, that's okay now.
No, it is clear, sir.
Sir, on the very left side from beginning.
From beginning, you go to beginning.
No, it is clear, sir.
Not clear, sir. You go to from beginning. There is an icon there.
Yes.
Just
That
below the file. Below the, yeah.
Yes.
No, it is clear.
Please
So you
click the icon from beginning.
Next target. No, it is clear.
Sir, you are not still on the slideshow mode.
What it is so problem.
You have to just click the from beginning. Yes.
No, it is clear.
I think you can continue with this because okay.
Why?
You can continue now. You can start.
My research topic is the suppressing effect of Conocarpus lancifolius aqueous extract physiology. Plant Conocarpus lancifolius, which is common name buttonwood, is an evergreen shrub or tree that belongs to family Cupritaceae and native to Florida mangrove forest ecosystem in North America. Its native habitat is moist and mostly grows on shoreline in tropical and subtropical areas around the world. Since the nutrient status of Conocarpus lancifolius is very low, so it grows very well in the soil that have very low fertility. Mostly it is planted in parks, streets, and yards as ornamental, and the potted plants are used to make bonsai plants. Tree release some phytochemicals in the soil which adversely affect the germination and yield of crops which are growing neighbor it. Cereals, also called grain crops, are generally defined as grasses grown for their edible seeds. The ancient Roman goddess for agriculture was called Ceres. Hence, the common food grains at that time were also recognized with her name. The word cereals is no change to cereals. The well-known cereal crops are wheat, maize, rice, barley, sorghum, oat, rye, and pearl millet. They belong to family Gramineae, also known as Poaceae. Economic importance of cereal. Cereal dominate the world food trade. Their stocks and prices reflect the major trend in the fluctuating world food situation. They are the economic source of protein and starch, contributing about 60% of calories in human diet. Wheat and barley are the important Rabi winter cereal, while the rice and maize are the major Kharif summer cereal. In some regions of Pakistan, millet, sorghum, bajra, and ragai are also grown. The important crops wheat, rice, barley, maize, and sorghum account 23.60% in the value addition of agriculture sector and 4.45% in the GDP. Any process involving secondary metabolite produced by plants, algae, bacteria, and fungi that influence the growth and development of agriculture and biological system is known as allelopathy. Allelopathy is an important mechanism of plant interference mediated by the addition of plant produced phytotoxin to the plant environment and competitive strategy of the plants. Allelopathy, any process involving secondary metabolites produced by plants, algae, bacteria, and fungi that influence the growth and development of agriculture and biological system is known as allelopathy. Actually, allelopathy is composed of two Greek words, allelion and pathos. Allelion means suffering, pathos means neighboring plant. Some plants have neighboring effects. Allelochemicals are released into the environment by all plant parts, that is, roots, stem, leaves, and fruit. In a specific environment, the allelochemical have enough strength to affect the neighboring plants. Interference may occur when one plant species fails to germinate, grows more slowly, shows symptom of damage, or does not survive in the presence of another plant species. Allelopathy is an important mechanism of plant interference mediated by the addition of plant-produced phytotoxin to the plant environment and competitive strategy of plants.
Dr. Hussain.
No, it is a flow
Can I
chart.
interrupt you?
No, it is
Yes.
a flow chart of a plant which
Dr.
shows
Hussain.
the allelopathic chemical release from the roots, leaves, stems, and due to the rain, these allelochemicals come to the soil and affect to the neighboring plants. All allelochemical releasing effects are shown by this picture. Stimulatory potential of crop water extract. Foliar application of 2% brassica, sunflower, moringa, and rice water extract promoted wheat grain yield significantly. Foliar application of sorghum extract has promotory effect on the growth and yield of crops as allelochemical present in it shows stimulatory behavior at diluted concentration. Most of the allelochemicals are water soluble and water is the best medium and carrier for such chemical. Water extract used to promote growth at low concentration.
Sir, sorry to interrupt you. I think your slides are not moving. You are at slide number 3 and please check.
No, I come to slide number, yes, slide number 3.
Yeah, you are speaking on slide number 3.
Your slides are not moving because you did not put your presentation on the slideshow mode.
Missing, no, this slide stimulatory potential of crop water extract is clear.
No, slides are not
This slide
moving.
is
Slide is not moving, sir. Yeah, now it is slide number 9, what I can see.
yes. No, I come to slide number 9, which shows the chemistry of these allelochemicals. It is clear, sir.
Yeah, it is clear, but you know, it should be changed while you are speaking. So for that it is
Comes to the slide number 9. Some allelochemicals involved in the allelopathy, in which these allelochemicals are releasing from the all parts of the plants, such as ferrolic acid, hydroxybenzoic acid, fumaric acid, synergic acid, cinnamic acid, purine alkaloids, and so many other allelochemicals like terpenses, glucosides, quinamides, aldehydes, phenolic compounds, open chain hydrocarbons, various secondary metabolites, all are involved in the allelopathy, and they are released from the plant parts either directly into the environment or may be transferred after decomposition of the plant parts. These chemicals affect the growth and development of the neighboring plants. See the some phenolic compounds: vanillic acid, salicylic acid, gallic acid, caffeic acid. These compounds are volatile in nature, but released from the living parts of the plants affect the neighboring plants. Now comes to the hypothesis of our allelopathy. Allelopathy in dilute or low concentration increase nutrient uptake and stress management, while allelopathy in high concentration cause insect disease and weed management, and we can use both of allelopathy in agriculture to promote our agriculture crops. Now comes to the material and methods side of our research. Postgraduate agronomy laboratory, Faculty of Agriculture, Gumal University, Deir Ismail Khan. Experimental design, completely randomized design. Application 4. Crops are oat, maize, rice, millet, wheat, and sorghum seeds sown in each disposable glass. There are 6 number of seeds are sown. Aqueous extract preparation. The leaves of Conocarpus lancifolius was collected, dried, and powder was made on grinding the leaves. The leaves powder with 25% weight by volume ratio was soaked in distilled water for 3 days at room temperature, and then aqueous extract was filtered, collected, and sprayed in a jar. Treatments: oat, amina fetua, treated as T1, maize, yamez, corn treated as T2, rice as T3, per millet as T4, wheat triticum estivum as T5, sorghum bicolor T6. Results: here we see the results of these cereal crops are affected by allelopathic effect of Conocarpus lancifolius. Control where only water application was done. Extract was used, and third is the difference between control and extract. The first table is about allelopathic effect of Conocarpus lancifolius on number of plants emerged or germination. The total number of 6 seeds were sown. Here we can see the different plants are affected by this solution. Majority of the plants affected positively, because in control their germination is low, but in extract their germination is high. This high germination may be due to the chemical present in the extract and cause release of some growth hormone into the seeds, which cause the germination of the plants. This is a positive effect, and later on its negative effects are also shown. Allelopathic effect of Conocarpus lancifolius on number of leaves. Here number of leaves of all crops are decreased in the extract as compared to the control. All the plants are depressed by application of this extract by Conocarpus lancifolius, mostly wheat is affected. Wheat and wheat, millet, rice are mostly affected by these application of aqueous extract, which release the allelopathic chemicals and cause the inhibition or suppression in all cereal crop. Now the table is third, allelopathic effect of Conocarpus lancifolius on fresh weight. Here we see also the suppression of the fresh weight of the plants emerged in all cereal as compared to the control with extract application. All plants have similar nature of traits. All are suppressed by application of these extract solution. Now comes to the allelopathic effect of Conocarpus lancifolius on dry weight. When fresh weight is also reduced, and this reduction also seen in their dry weight accumulation, here we also see the negative impact of these extract on all the cereal crops when we compare the control with the extract obtaining the different dry weight of the plants. All cereals are mostly affected by application of this extract. Now we come to the plant height. Allelopathic effect of Conocarpus lancifolius on plant height. When we compare the value of control with the extract, oat plant seem as positive, oat, millet, and sorghum. Both these three plants are C4 plants and C4 plants and desert plant. They show positive towards the Conocarpus lancifolius extract. Remaining cereals show the negative impact on their growth and plant height when we applying the Conocarpus lancifolius extract. So comes to the allelopathic effect of Conocarpus lancifolius on shoot length. Shoot is that portion which emerge from the soil. Here we see also the similar result as in the plant height. Oat, millet, and sorghum have positive impact, while remaining three cereal have negative impact on the shoot length. When we applied the extract solution on these plants, they are suppressed, and remaining three C4 plants, which are also desert plant and can grow in the arid area, are mitigating the effect of Conocarpus lancifolius, but our major crops are mostly affected by this extract. Allelopathic effect of Conocarpus lancifolius on root length. Here also shows a similar result as the previous slides. Oat have positive effect, but remaining crops are badly affected by applying Conocarpus lancifolius effect, while maize have no effect on root length when we apply the Conocarpus lancifolius plant extract in the field. It will show that maize is resistant to somewhat by this extract, but remaining crops except oat all are affected negatively by applying Conocarpus lancifolius effect. Allelopathic effect of Conocarpus lancifolius on cotyle length. Cotyle is that non-green portion which emerge from the soil and cause the epicotyle and plumule to convert into shoot. This cotyle length also shows that mostly plants are positive or negatively affected by these plants. Oat, millet, sorghum, these three plants are positively affected by this extract, but remaining crops, maize, rice, wheat, or negatively affect their cotyl length. Allelopathic effect of Conocarpus lancifolius on chlorophyll content. Here, all crops are badly affected by applying Conocarpus lancifolius extract. Mostly the maize crop is too much badly affected, where chlorophyll content dropped up to 7.5 microgram per centimeter square as compared to control. Overall, all the plants are affected by applying the Conocarpus lancifolius uh, extract. Conclusion: Conocarpus lancifolius extract 25% weight by volume, mostly having adverse effect on most parameters studied except plant emergence. However, each cereal crop response is different in different parameters. For further research, it is suggested to evaluate the suppressing effect of other parts of Conocarpus lancifolius like its bark and root to further confirm its allelopathic impacts. No, thanks. My presentation is over. No house is open for the questions. No house is open for the questions. My presentation is over.
So, thank you, Dr. Iqtidar Hussain, for your nice presentation about the allelochemicals and allelopathy and allelopathic effects on the different plants. Well, due to shortage of time, it is not possible to take the questions on. So, once again, thank you very much. And now we move to the next speaker. The next speaker is Dr. Pankaj Gupta, Assistant Professor,
Thank you,
Department
sir.
of Chemistry.
Thank you, organizer, for
Thank
making you an opportunity.
Thank you, sir. Thank you, sir. Thank you so much. So next speaker is Dr. Pankaj Gupta, Assistant Professor, Department of Chemistry, Government Degree College, Kathua. He is going to speak on Arthrobacter species lipase, a versatile biocatalyst for the synthesis of enantiopure compounds. So, Dr. Pankaj, you can now share the slide,
good morning, sir.
and you can start now without wasting any time.
Good morning, sir. Good morning. Highly esteemed chairman of today's session and my Robert teacher, Professor S.K. Pandey, sir. Convener of the conference, Avinash, sir, my teacher also. So, very good morning to all of you and distinguished speakers.
Thank you.
So, no, without wasting time, I'm going to share this. Okay, no?
Yeah, now it's okay. Yeah, good.
Okay, thank you, sir. Thank you. So, the title of my speech is Arthrobacter simplex species lipase, a versatile biocatalyst for the synthesis of enantiopure compounds. So, as you know that chirality is not a prerequisite for bioactivity, but in bioactive compounds where a chiral center is present, great differences are usually observed for the activities of the enantiomers. So, this phenomena is usually observed in natural compounds as well as the synthetic compounds available in the medical store. So, whenever a compound consists of one chiral center, the two enantiomers will be there. The one will be called the eutomer and other will be called as a distomer. The distomer is the less potent molecule and the eutomer is a more potent enantiomer. So, single enantiomer drugs would provide superior therapy by allowing reductions in dosage, reduced variability in metabolism, simpler dose response, and improved tolerability. So, there are some compounds why we are going towards the chiral, that is single enantiomer drugs. So, actually, enzymes, receptors, and other binding molecules in biological process can recognize enantiomers as different molecular entities. So, out of top 10 drugs, 9 contains the chiral active ingredients, and for 6 of them, this ingredient is a single enantiomer molecule. So, 70% of the chiral drugs are marketed as racemates, and in 2020, the 20 out of 35 pharmaceuticals approved drugs are chiral. So, now we are going towards the single enantiomer drugs rather than the racemates. So, from 1992 onwards, this process towards the single enantiomer drugs is going on enhancing. So, this important trend towards a single enantiomer drug is what has brought asymmetric synthesis to the forefront. No, our main motive is to go towards the asymmetric synthesis, or changes the racemic drugs from the removing the racemic drugs from the market, and bring the new drugs that are the pure enantiomer drugs in the market. So this is the receptor drug interaction. Here we can find that the epinephrine has two types of that is enantiomers. One in which the OH side here, for clarity, it is shown down, and the OH is the first is shown up. These are two different enantiomers. When in the second enantiomer, it binds with the receptor and show its activity more as compared to the earlier one, which is in which the OH group is upper side. So the pharmacokinetic kinetics, metabolism, and the toxicity and the immune response are mainly responsible for the difference in the activities or biological response of these enantiomers. So this is the FDA dossier. So Food and Drug Administration in 2015 approved various chiral drugs, and out of these all are chiral, but out of these only one named Lesuner, which has been licensed as a racemate because of the atrope isomerism presence and the restricted rotation around the carbon-nitrogen bond. So no, I will tell some of the drugs which are available in the market and show different biological activity of the enantiomers. This is the Nephroxane. So Nephroxane is given as an anti-arthritic drug, but its Nephroxane is very potent, very active, and whereas the R enantiomer causes the liver poisoning. So that is why in order to reduce the side effect of the other enantiomer, the only one enantiomer is required, and the other should be removed from the market, and no, it is removed also. So the second is thalidomide. It is given to the pregnant woman in 1960s in USA for the treatment of morning sickness, in order to avoid morning sickness. But what happened in the later on, the newborns, babies, were teratogens without limbs. So that is why that drug is banned in the USA in 1965, and then 1996 or onwards, it is again available in the market, but for different purpose, not for the pregnant woman. So its R enantiomer is distomer is a sedative, while S enantiomer is highly teratogenic. So H enantiomer should not be given to the pregnant woman. So this is another molecule, that is ethambutol. It is an anti-tuberculosis drug. So it has been found that the SS ethambutol is 200 to 500 times more potent as compared to the RR enantiomer. So this is another penicillamine. S-penicillamine is anti-arthritic, while the R enantiomer is a mutagen. So it is petiol. It is the pheromone component, and the one diastereoisomer is the male-specific attractant, whereas the other as identified as an aggregate phenomenon. So this is fluoxetine. It is also very important antidepressant drug, and it has been found that the S enantiomer of fluoxetine does not cause adverse side effects, whereas the R enantiomer causes the side effects. So in order to reduce the side effects, the one enantiomer should be given to the patients rather than the racemic. So it is salbutamol. It is now available in the market as a nebulizer. No, most of the people, they are this COVID-19 situations, they are using as a nebulization ampoules. So its one enantiomer is active, whereas the other enantiomer is responsible for the unpleasant side effects. So that is why it is now available as a single enantiomer in the market. So citrazine. Earlier we are going to the market and take citrazine as a tablet, but no, we ask the medical practitioner to give the levocitrazine, and the dose has been reduced from the 10 milligram to 5 milligram. So that is why the single enantiomer drugs have a lot of applications as compared to the racemates. So these are the different methods for the synthesis of enantiomerically pure compounds. We can prepare the enantiomerically pure compounds from chiral pools, from racemates, from prochiral substrates. But in our present approach, what we applied in our synthesis is the biocatalytic synthesis. So biocatalytic synthesis, the basis of biocatalysis is already discussed yesterday by our keynote speaker, that is Professor Anju Chadda ji. So she beautifully explained the basis of what that is called biocatalysis. So in our present approach, I will show only the application part because of less time. So application time, we are using enzymes for the preparation of enantiomerically pure compounds. So over the last 3 days, an explosive growth of research in the field of biocatalysis has taken place. So what we are doing here, we are integrating biocatalysis with the modern chemical research, so in synthesis, so that the stereoselective and regioselective synthesis can be done. So during the screening program in IIM Jammu, we are successful in identifying a lipase that is called the Ortho Simplex lipase, and that is found out to be the superior as compared to the other lipases that were commercially available, as well as the indigenous lipases that were screened during that period. But till now, the Ortho Simplex lipase that has the MTCC number 15125, so it is superior as compared to the others, other commercial as well as the indigenous lipase for some of the pharmaceutically active ingredient preparations. So this is the extraction or the purification procedure for the preparation of for the isolation of Orthobacter species. Here, when we have taken the cell biomass, the activity is 1 unit per milligram. After isolation, we got the 17 unit per milligram of the protein. But when we do the precipitation and the cell-free extract ammonium sulfate precipitation, after that, the activity is 51.7 unit per milligram, and at last Saffrose cell filtration, we'll get the 655 unit per milligram, and at the last Cefadex, we get the 1700 unit per milligram of the protein. So after that, we applied this we use the immobilization technique also to get the enantioselectivity factor to enhancement or the modulation of the enantioselectivity or the stereoselectivity. These are the different sports that we applied for the immobilization of that particular lipase that we prepare, that we isolated. So this is the magnetic sol gel, non-magnetic sol gel, poly, and GNP or ANP type sports. So now come directly to the application part, where we applied this lipase for the hydrolysis of different substrates. We use the desymmetrization techniques also. Here, after desymmetrization techniques, desymmetrization means here the molecule is symmetric, but after the removal of one R group, the molecule becomes asymmetric and contains the chiral center, and the enantioselectivity that we got is greater than 65 and up to 74%. That is not so good, but it is applicable for desymmetrization also. So here, again, we have taken the different phenyl ethanol substrates and the esters. From that, we also got the enantioselectivity up to 99%. That's the target value for the enantioselectivity or the work. When we start some work, we especially put a target value of greater than 99% enantiomeric excess. And in these substrates, we got. So now we come to the 1,2-amino alcohols, which are widespread structural motif in natural and synthetic biological active molecules. So these are some of the drug molecules that are available in the market. And our target is to prepare some of these drug molecules in the enantiomerically pure form. And in somehow we were successful also. So these are the first is the bistatin, adjuvant in cancer chemotherapy. Second is the propranolol, it is anti-hypertensive drug. Or timbamide, it is hypoglycemic activity showing. Or agilene, hypoglycemic activity. Paclitaxel, anti-tumor agent. Dinopamine, sphingosine. So we started with the coupling of potassium phthalimide with the bromoketone, alpha bromoketone, and after successful reduction, that is palladium carbon reduction, we are getting the alcohol, and then after acetylation, we put the kinetic resolution strategy. We used kinetic resolution strategy for the resolution of this particular 22 number substrates, that is the thalidoacetates. And we are successful in obtaining the product in greater than 99% enantiomeric excess. Similarly, the above strategy is applied for the aliphatic substrates also, and here also we are able to get the enantiomeric selectivity up to 99%. So these are some of the Hplc profiles where we are in the first, it is showing the racemic. The retention time is 22 and 30. In the second, it is again racemic, and then the retention time is 34 and 37. But in the last one, it is showing the enantioselectivity of the substrate as well as the product. Here, the first one is 22 is there, but 30 is absent. And in the second one, 34 is present, but 37 is absent retention time. So that means that we are able to get the products with the 99% enantioselectivity. So similarly, the other substrates also where we are able to get the enantioselectivity that is greater than 99%. So there is another type of substrates that is called the Aryloxy propanolamine. These are beta adrenergic blocking agents. So beta blockers, we are able to prepare beta blockers also that are in greater than 99% enantiomeric access. So this ABL catalyzed enantioselective resolution strategy is also applied for the cycloalkanols, that is cyclo 2 alcohol 1 thaliamido. So here also we are able to get enantioselectivity greater than 99% when we use ABL, but in biphasic system. In all the cases, when we use a kinetic resolution strategy, we use the biphasic system, that is Tolvin buffer system, for the accomplishment of this particular enantioselectivity. So these are the different enantiomerically pure compounds that we prepared in the due course of time, and with a good enantioselectivity, that is up to 99% enantioselectivity. So this is another very important molecule that is called the sucralose. It is it is used in more than 28 countries as a sweetening agent. So it is taken by the diabetic patients as a sweetener, artificial sweetener. So currently about 120 products that are involving sucralose as a sweetener are in the US market. So by using ABL, we are successful in regioselective conversion of sucralose 6-acetate to sucralose. So that is a very important project, because the chemical methods are a multi-step techniques, and the biocatalytic methods are also not good that were reported in the literature. But in our case, ABL was successfully applied with the 100% conversion and the no purification required at the end. So this is another application of our Orthobacter simplex lipase, where we are successfully resolved the atrope isomers. Atrope isomers are the isomers which show the restricted rotation around the carbon-carbon bond. So when we take the racemic vinol is a very important catalyst that is used for the carbon-carbon bond generation. That is enantioselective carbon-carbon bonds generation, Friedrich-Torf reaction, Bellis-Hellman reactions, or various enantioselective meso-epoxides ring opening reactions. Here it is used as an enantioselective catalyst. So we are successfully able to obtain the product in greater than 97% enantioselectivity. So here also there is another molecules that are called Alkoxy, Aryl acetaldehydes, or acids. So these are also very important molecules, because they can be used for the determination of absolute configuration of secondary alcohols. And these can also be used as a plant growth regulators, calcium antagonists, and some as a apoptosis inhibitors. So we are successfully able to prepare all these molecules and their final products with the greater than 99% enantiomeric excess of the product. So this is another important strategy where we are able to prepare 4 different drug molecules that are called prozimpal, piperosine, and dibosine, and doxazosine. The first 3 molecules, that is prozimpal, piperazone, and dibosine, are alpha adrenergic blockers, whereas the doxazosine is used as an anti-hypertensive drug. So by using the kinetic resolution strategy, we are successfully in preparing these 4 drug molecules in good enantioselectivity. And no, at the last, before using much time, I am going to conclude this topic. So conclusion is that the development of new chiral separation techniques is and will be the topic of subject in academic research as well as in the industrial advance. So by using the biocatalytic processes, the products that we get is the high what that is called high yield products, or you can say that they are valuable products. These are considered, because these are considered as compared to the natural products. So ABL, our Orthobacter simplex lipase, is considered, or it is proved to be the better lipase or enzyme as compared to the commercially available pharmaceutical, you can say, for preparation of pharmaceutical intermediates. So moreover, in some cases, the immobilization of ABL was also carried out, and it has been found that after immobilization, it can be reused for several cycles without any significant loss of activity. So enantioselectivity as well as the regioselectivity can be obtained by immobilization of this ABL on magnetic as well as the non-magnetic spots. So at the last, I want to say that the Orthobacter simplex lipase will continue to be an excellent biocatalyst for preparing enantioselective compounds. So in the near future, biochemical techniques and the innovative experiments are required in cellular and molecular biology, which can prove to be a real breakthrough in commercialization of this APL. So with these words, these are some research outcomes, and thank you, sir, and thank you all. Now